Hi there, my name is Dr. George Murphy. I'm an assistant professor in medicine in the Division of Hematology Oncology at the Boston University School of Medicine. And I'm also one of the co-directors of the Boston University and Boston Medical Center's Center for Regenerative Medicine, which we affectionately refer to as the CREM. Basically, we're a bunch of sort of like-minded researchers that are interested in all things stem cell biology and regenerative medicine related. More specifically, what I do and my group does is we're very interested in understanding exactly how blood cells form in the human body, and even more specifically, maybe when this process goes wrong, what actually happens, such as in diseases like sickle cell anemia, where red blood cells form improperly and then leading to all sorts of negative downstream consequences in many different organ systems. And we generally explore these areas of research using really cool stem cell-based models, viruses and vectors that allow us to insert genes into cells themselves, and other really sort of high-tech cell and tissue culture-based systems that really allow us to explore and investigate all these questions that we have. I suppose I'm a bit of an anomaly. So whereas many people that actually get into medicine and science maybe come from a background of affluence, I never really knew who my father was. I grew up separately from my mom, and I grew up in really sort of depressed cities that were really sort of bad neighborhoods. So for me, knowledge wasn't just power. For me, it represented a way that I could sort of escape this life that many of my peers thought was hopeless. And the way that I did that was through sort of a lot of hard work, a lot of understanding of what the situation was, a lot of luck, and being able to harness a great number of opportunities and scholarships that allowed me to train at some of the best schools in the world and really live out my hopes and dreams. What everyone, I think, in medicine and science would aspire to is to change the world, and we really believe that. And if you talk to any of the trainees that are actually here, they drink the Kool-Aid as well. We're all trying to change the world. In the short term, some of the things that we're actually doing right now, today, those stem cells that I told you about, those pluripotent stem cells that can become any tissue type, we have the ability to specify them into many different cell types that are found within the body. And what we're doing right now is something we refer to as the clinical trial in a test tube, or CT3 for short. And what this allows us to do is we can also make these specialized stem cells from patients with various genetic illnesses. And we can then take those stem cells and specify them into the tissue type that may be affected in that particular disease. Much in the same way, as you're going through that process of specification to these cells, we can actually investigate the disease processes that develop during that process of development. And at the same time, this process of the clinical trial in the test tube allows us to test maybe novel therapeutics or novel treatments in the exact genetic context of these patients. So in essence, what we can create are patients or subjects that are actually in a dish that may have a particular illness or disease, and model that particular illness and disease, and at the same time, perhaps develop drugs that could actually be used to intercede in the process of that disease development. And that's something we're actually doing right now. Every day for me is different. So one day I might be strategizing with my people, designing the next set of seminal experiments that might be done. We might have fantastic collaborators that are actually coming in to discuss a new phase of science that we haven't even embarked on before, or I might be traveling to a scientific meeting across the world to learn about the newest and most exciting science that's coming about. I also really like to be in the thick of things. I like to be elbow to elbow with my people at the bench and in the tissue culture hood, because there's nothing more exciting than setting up your own experiments and then coming in the next day to see what's actually happened, maybe observing something that no one's actually seen before. My mom was a fantastic artist, and I definitely have artistic qualities that come from her. This is actually a painting that I did. It's a faceless samurai. I kind of think that in some sense we see ourselves as scientific samurai, kind of doing things for the benefit of humankind. And so art, I think, is really underestimated, as is creativity and science as a whole. So I really like to apply creativity in my science and the design of my experiments. So for me, I'm definitely much more creative than I am technical. And I think that's important. So I think if there are young students that are sort of coming up in the game that think that they are far more creative than technical, maybe don't have the kind of left brain required for mathematical thinking, that in no way should deter them from a career in science or technology, because creativity is an essential aspect of the equation as well. In essence, what I would say, what we do is we actually get paid. We have the greatest job in the world because we actually get paid to figure cool stuff out. That's what we do.